Welcome back, friends. Good evening. So in this session we are going to see about the biochemical characterization of the transforming principle. So last session I had told you about the transformation principle. Who performed that experiment? Griffith. So Griffith, what he said, some factor is there, some principle, transforming principle is there from one bacteria to another bacteria, that only transmitting the characters. That is why the bacteria has been changed into a virulent bacteria. So that is the concept in the previous session. Here what they are going to do means they are going to characterize the biochemicals, biomolecules present in the estrine which is heat killed. So in that experiment, the previous Griffith experiment, what he did, he was killed the estrine, that is the virulent strain. You can see here, this is the estrine, virulent strain. It produces the mucus layer over the cell wall. Okay, so this mucus layer acts as a toxin. So this only creates a pneumonia in organisms and leads to the death of the organism. So next what he did, he heat killed the estrine. After heat killing, then he mixed with the R strain. After R strain, what will happen? The mice has been dead actually. Okay, so this is happened in the last experiment. So these three scientists, what they did means this Oswald Avery, Colin McLeod, Maclean McCarthy. So these three scientists only took the Griffith experiment. From that they did the biochemical characterization. So up to this experiment, all the scientists were thinking that protein is the genetic material. So protein only transmitting the characters from one generation to next generation. So that was a thought during that time. So after this experiment, what happened? Everything has been changed. So what happened here? Let's see here. So you can see here, this is what estrine, that is virulent strain. You can see here the mucus layer is there. When it is heat killed, what will happen? As I said, right? Cell wall will break down, cell membrane will break down. For example, cell wall, cell membrane is broken down. What will happen? Full of lipid molecules will be there. Suppose for example, next what will happen? The mucus layer is broken down, so polysaccharides will be there, and then inside many proteins will be there, RNA will be there, and DNA also will be there. So after heat killed, what will happen? You can see here the mixture of the heat killed estrine consists of lipids, RNA, DNA, proteins, and many other things will be there. So here majorly you can look into only these four things. So lipids will be present in the mixture, as well as RNA will be there, DNA will be there, and proteins will be there. So this was first isolated by these three scientists. So what are the biomolecules present in the estrine mixture, heat killed mixture? So after this, what they are doing, without the treatment of any enzyme, so you are not treating with any enzyme, what they are doing, simply they are adding this with the R strain. So without the treatment of the enzyme, they are mixing with the R strain, that is a live strain. Then they are injecting into the mice. Now what happens? The mice died. Why? Because one of the factor from here transferred to the R strain. For example, it may be lipid, maybe RNA or DNA or protein. Among the four, anything may be transferred from here to there. So one of the thing has been transferred from here to there. So it transformed the R strain into virulent strain. That is why it caused disease. So the mice died. Now what they are doing? They are going to treat with specific enzymes. Okay, degrading enzymes. Then they are going to check. So the heat killed estrine, so consists of this mixture, has been treated with, that is mixed with what? R strain. Now what they are doing, this heat killed estrine plus R strain is treated with the enzyme called as lipase. So lipase, what is the function of the lipase? So that will degrade the lipid molecules present in the mixture. So here lipids are there. So these lipids will be broken down, they will be degraded. Now this heat killed estrine consists of what? Only RNA, DNA and protein. Only three contents are there, lipids are not there. Then they are injecting into the mice. Then also what happened? Mice died. What denotes here? So the lipids are not responsible for transformation. Suppose in case lipids are responsible, means what would be the result? Mice will be alive because lipids are not transformed here, because lipids are degraded in this case. So here lipids are degraded, lipids are not there, only RNA, DNA, proteins only injected into the mice. Now the mice died. So what will be the result? Among these three, it may be RNA, DNA or protein. Among these three, anyone is responsible for the transformation. Again, what they are doing? Again, they are treating with RNase enzyme. So, RNase enzyme, RNase what it will do? It will degrade the RNase. Now, what will happen? Now, the RNase will be degraded. Next, if you see here, this RNase will be degraded. So, what will happen? RNA will not be present in the mixture. Now they are injecting into the mice. Again the mice died. Now what will be the result? So RNA is not responsible for transformation. Suppose if it is responsible for transformation, mice would be alive, but mice is died here. So RNA is not responsible for the transformation. The next thing what they are doing? They are treating with protease enzyme. Now when they are treating with protease enzyme, what will happen? These proteins will be degraded. Okay, now what will happen? Remaining all the three things will be there. DNA will be there, RNA will be there, lipids will be there. They are treating with only protease. So all the proteins will be degraded. Now these three contents are injected into the mice. Now also what happened? Mice died. So proteins are not responsible for transformation. At last what they are doing? They are treating with DNAs. DNAs what it will do? It will be going degrading the nucleic acids called as what? Deoxyribonucleic acids. So all the DNA will be degraded now. After the degradation of the DNA, now the mixture consists of what? Only lipids, RNA, proteins. Only these three will be there. DNA will be absent in the mixture. Now when this is injected into the mice, what happened? Mice is alive. So from this experiment it is clear that when DNA is not there, mice is alive. So it is the transforming principle. So DNA is the thing which is transforming the normal bacteria into the virulent bacteria. So DNA is responsible. So you can see all through these four experiments. Here also DNA is present. So mice died. In this case also DNA is present, mice died. Here also DNA is present, mice died. Whereas here DNA is absent, mice is alive. That means wherever the DNA is present, the mice died. Wherever the DNA is absent, mice is alive. So DNA is a transforming principle. So DNA acts as the genetic material. So that is the conclusion given by these three scientists as a result of the biochemical characterization. So in this case, lipids are not responsible, RNA is not responsible, proteins are also not responsible. Only DNA is responsible. We will meet in the next upcoming session. Thank you, friends.